Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CSIR Net JRF Life Science question paper. This is the shift 1 question paper. I am discussing the part C questions. This was conducted in March 2025. I will be discussing the cell biology questions. So coming to the first question. Protein X can be extracted from disrupted erythrocyte plasma membranes with high salt concentrations. Okay. So how is protein X extracted? Usually it is extracted from, you disrupt the erythrocyte plasma membrane, treat it with high salt concentration, protein X is extracted. Now, treatment of intact erythrocytes with protease followed by extraction led to intact protein X. Treatment of disrupted erythrocyte plasma membrane with protease followed by extraction led to fragmented protein X. Now don't get scared. First time when you are reading what is this, you will feel. But read the interpretations and you will come to know what the examiner wants from you. So first they have given protein X is peripheral and in B it is integral membrane protein. Then they have told protein X is on the extracellular matrix phase of the plasma membrane, and in option statement D they have told it is on the cytosolic phase. So here what you have to know is the examiner wants to know is whether protein X is peripheral or integral, and whether it is on the extracellular phase or on the cytosolic phase. That is why two experiments have been conducted here. One is they have disrupted the erythrocyte plasma membrane, treated with high salt concentration, and protein X got extracted. From that statement itself, you can come to know whether it is a peripheral membrane protein or an integral membrane protein. So what is peripheral membrane protein? Found on the plasma membrane. It is attached by electrostatic interactions, and it is found on the surface of the plasma membrane, and usually it can be extracted by high salt concentration. And integral membrane proteins, they are found in the lipid bilayer, and first what you have to do is you have to disrupt the lipid bilayer by treatment with detergents, and then you have to isolate the proteins. That is how you isolate integral membrane and peripheral membrane proteins. So I told you peripheral membrane protein isolated with high salt concentration. So A is the correct answer and not B. Okay, then you have to see for C and D. So out of these, 3 gone, 4 gone, you have to see 1 and 2. Now B is wrong. Okay, now C. C and D, they have asked whether it is on the extracellular phase or on the cytosolic phase. Now see this figure. This is your plasma membrane. This is the extracellular side. So if the protein is here, extracellular side. If the protein is on this side, then cytosolic side. This is the plasma membrane. Here is your cytoplasm. So this is the cytosolic side. This is extracellular side. Okay, to know whether it is on the extracellular side or the cytosolic side, what you did was, or what the experimenter has done was, he took intact erythrocyte membranes. Okay, intact erythrocyte, no disruption of the plasma membrane, and he treated with protease. Okay, he treated with protease. Protease, what it will do? It will fragment your protein into pieces. Okay, so treatment of intact erythrocyte with protease followed by extraction. Then he extracted the protein using high salt concentrations. And what did he get? He got intact protein X. There was no disruption of the protein. So if the protein was on the extracellular side, upon treatment with protease, it would have got fragmented. And once you extract it with high salt concentration, you are not going to get an intact protein X. Okay. Now, if the protein is on the cytosolic side, it will be protected from protease because it is on the cytosolic side and intact erythrocyte have been used. So it will be protected from protease and you will not get a disrupted protein X. Okay. Now, next what he did was he treated the disrupted erythrocyte plasma membrane with protease followed by extraction led to fragmented X. Now what he did, he disrupted the plasma membrane. Okay, made a plasma membrane into pieces. Then he treated it with protease. Okay, treated the disrupted membrane with protease. So your protein which is on the cytosolic side got disrupted because of protease treatment. Plasma membrane is also disrupted. That means the protein present on the cytosolic side got exposed outside. It came into contact with your proteases and got fragmented. And then he extracted it and he got fragmented protein X. So that shows the protein X is on the cytosolic phase of the plasma membrane, not on the extracellular. So when he used intact erythrocyte protease treatment, he got intact protein X upon treatment, upon extraction. But when he treated disrupted erythrocyte, made plasma membrane into pieces, followed by protease, this got exposed, it got disrupted with the protease and you got fragmented protein X. So that shows it is on the cytosolic side. So that makes option C wrong and option D correct. So answer here is option 2. 1 is not correct. Then spindle assembly in animal cells requires nuclear envelope breakdown, NEBD. Okay, this is happening in your mitosis. So NEBD is a multi-step process which begins when CDK1 cyclin B phosphorylates multiple components of the nuclear envelope. So nuclear envelope breakdown happens when CDK1 cyclin B, it will phosphorylate certain components on your nuclear envelope. So given below are some components that are directly phosphorylated by CDK1 cyclin B. You have to choose the correct CDK1 cyclin B substrates that are directly associated with NEBD. Okay, NEBD is nuclear envelope breakdown. So nuclear pore complexes, yes, phosphorylation, it will result in a nuclear envelope breakdown and they are phosphorylated by CDK1 cyclin B phosphoryl enzyme. So A is there. Then nuclear lamina, of course phosphorylation by CDK1 will lead to disassembly of the nuclear lamina during nuclear envelope breakdown. That's also correct. Now great wall kinase, yes, that is also phosphorylated and activated by CDK1 and you know it, what great wall kinase does is it inhibits the phosphatase activity of PP2AB55, another enzyme, and it promotes mitotic entry. So CDK1 phosphorylates great wall kinase. But as per the question they have asked which one is directly associated with nuclear envelope breakdown. Great wall kinase doesn't have anything to do with nuclear envelope breakdown. So that cannot be the answer. Anyway, they have not given A, B and C here to confuse you all. So C is not the answer. Then histone 3. Okay, histones are found in the chromatin structure. So phosphorylating histone H3, it doesn't make sense with regard to a nuclear envelope breakdown. Okay, so D is also not correct. So A and B only, that makes option 3 as the right answer. Okay. Then next one. Actin dynamics in cell is dependent on several actin binding proteins. Given below are actin binding proteins in column X and their function in column Y. You have to match the following. So actin binding protein, they regulate the structure and function of the actin. And there are so many actin binding proteins. Some are involved as nucleation factors. That is, they help in the assembly of new actin filaments. That is nucleation. Then some actin binding proteins, they bind multiple actins together. They bring together multiple actin filaments together and they can create either bundles or they can form networks. So such kind of actin binding proteins are also there. Then some help in linking actin filament to the cell membrane. So like that, so many actin binding proteins are there, and 4 of them they have given, and you have been asked to choose the function of each of these. So formine it is a help as initiation or nucleation of new actin filaments, and they remain attached to the growing end of the actin filament and help in continual growth of the actin filament. So that is what formine does. So that makes formine as 3. Okay, A3, that means 1 and 2 gone, 1 and 4 gone. 2 and 3 we have to see. So A3 is there in 2. So nucleates assembly and remains associated with the growing plus strand. Then ARP23 complex. So ARP23 complex, it helps in the assembly of actin filaments and it creates a branched network. So B1, that makes option 2 as the right answer, because in 3 it is B2. So B1A3. Then coffulin. Coffulin, it promote depolymerization of actin. So it is okay. For C4 they have given in all the 4 options. Okay. Binds ADP actin filament, accelerate disassembly. Then tropomyosin. Okay, you may be thinking what it is doing in actin filament. It is involved in contraction. Okay, not only that, it has action in actin filament. It is also one of the actin binding protein, and what it does is it contributes to the stability of the actin filament, and it influences the binding of other actin binding proteins to actin filaments. That is what tropomyosin is doing here. So D2, it is stabilized filament, modulates binding of other accessory proteins. So D2 is the answer. So answer is option 2. Then next one, given below are different types of bacterial growth curves. You have to say which is continuous, which is asynchronous, which is synchronous, and which is dioxic. Okay, so if you see the first one, you can see that it exhibits a zigzag pattern, and here the cells divide roughly at the same time. Then there is a period of rapid population growth, then period of no increase, then again a rapid population growth, then no increase, then again rapid population growth, then no increase in the number of cells. So this is called as synchronous bacterial growth. So that makes option 1 and 3 out, and 2 and 4 we have to see. Then next one, B, it is asynchronous. It is having the lag phase, log phase, death phase, everything is there. So it is asynchronous, so that means option 4 is going to be your correct answer. Okay, we will see the others also. Then continuous. Okay, continuous means you keep adding fresh medium and you constantly remove the old medium. So this will help in maintaining the nutrient supply and waste removal nearly constant. And because of this, it allows the bacteria to remain in the exponential stage of growth, and it can divide indefinitely. So it will not experience the stationary and the death phases. So that is seen in continuous growth. So C is continuous growth. Then dioxic. Dioxic is if you add two different sugars to the medium. That is, one you add glucose and lactose. Bacteria, you all know, it is having more preference towards the glucose than the lactose. So it will start utilizing the lactose and it will increase, and then glucose will be depleted from the medium, and the bacteria will go to a slowing down phase of growth. So that it will prepare itself to start utilizing the second sugar, that is the lactose, and this is the lag phase. Then there is the exponential phase where it is utilizing lactose and beginning to grow again, and finally the stable phase. So that is seen in the case of dioxic growth. So 4 is synchronous, B is asynchronous, C is continuous, and D is dioxic growth. Then next one, okay, given below are the components that facilitate transfer of molecules across phospholipid bilayers. So these are the membrane components and these are the activity you have to match. Then ATP powered pumps, okay, they move substances against the concentration gradient or the electrochemical gradient. So that is seen in A3. Movement of molecules against a concentration gradient or a electric potential. So A3, that makes 1 and 2 out, 3 and 4 we have to see. Okay, then uniporter. Uniporter also, it doesn't require ATP and it moves only a single type of molecule across the membrane from an area of high concentration to an area of low concentration. The first one is against concentration, no. Molecule against concentration, no. Transport of hydrophilic molecules down their concentration gradient, that is from higher concentration to lower concentration. So that is in the case of B. So B is 4, okay. Again competition between 3 and 4, they have given B4. Now channels. Channels also, they allow molecule to pass through by facilitated diffusion. So C, yes, and it doesn't require ATP, that is they move down their concentration gradient. So you can say C will be to free diffusion of hydrophilic molecules. That is what channels does. So C2. Then symporter. Symporter, if you take the case of sodium and glucose transport, you can see that sodium moving down their concentration gradient and they provide energy for the glucose to be transported into the cell against the concentration gradient. So sodium down their concentration gradient, glucose against their concentration gradient, and they move in the same direction. So if you see one movement of molecule, again concentration gradient coupled to another molecule moving down the concentration gradient. So D1. So that means option 4 is the right answer. Then, okay, now before coming to the next question, just we will have a look at all these things. So there are 4 major types of coated vesicles that are involved in the transport between various organelles or plasma membrane in the cell. One is clathrin coated, the green capped ones, COP1 coated, that is the blue capped ones, and COP2, that is red capped ones, and there is the 4th type called as the retromer coated. It is not shown in the figure, I didn't get a picture for that. Okay, there is the 4th one, that is retromer coated. So Clathrin coated, they mediate transport originating from the Golgi apparatus to the endosomes and from there to the plasma membrane. Okay, that is what Clathrin coated vesicles are doing. Now COP1, that is help in the transport originating from Golgi cisternae. This is the Golgi cisternae, from there to endoplasmic reticulum. And COP2, it is involved in the transport originating from endoplasmic reticulum to the Golgi. That is what COP2 is doing. And what is retromer? It is mediating the transport of protein from endosomes to trans-Golgi. That is this way, from endosomes to Golgi. From endosomes to Golgi. That is what retromer coated vesicles are doing. Now again, before coming to the question, I have one more concept, that is KDEL receptor action. So what is happening here? So this is the endoplasmic reticulum. You have, it is synthesizing proteins, and proteins are moved from endoplasmic reticulum to Golgi through COP2 coated vesicles. Okay. So if in case a protein which is destined to be present in the endoplasmic reticulum by mistake got transported to the Golgi complex, then what will happen? So proteins destined to be present in the endoplasmic reticulum will have KDL sequence in them. So this KDL sequence will be recognized by the receptor KDEL receptor on the Golgi, and it will recognize this KDL sequence peptide. It will package it into COP1 vesicles and send it back to the endoplasmic reticulum. Okay. So COP2 endoplasmic to Golgi, COP1 Golgi to endoplasmic reticulum. Okay, this is what is happening. Now read the question. Given below are few statements about intracellular protein transport. Proteins that are destined for the lysosome are tagged with mannose-6-phosphate group in the Golgi apparatus, which is recognized by the M6P receptor in the trans-Golgi network. That is 100% correct, and this has been asked previously in CSIR. The same question have been asked. Okay, so that A is correct. Then signal recognition particle directly mediate the insertion of protein into the mitochondrial membrane. There is no SRP or signal recognition particle in the mitochondrial membrane. It is present in the endoplasmic reticulum, not in mitochondria. So that makes option B as the wrong answer. Then KDL receptor in the endoplasmic reticulum and Golgi apparatus work by retrieving soluble endoplasmic resident proteins that have accidentally moved to the Golgi. Yes, that is correct. I just now explained that concept. Then cargo proteins that need to be exported from the endoplasmic reticulum are packaged into COP2 vesicle based on the presence of an endoplasmic reticulum export signal in their cytosolic tail. Okay. Now these proteins which are synthesized in the endoplasmic reticulum, they have an export signal in their cytosolic tail. Now such kind of proteins, they are packaged into COP2 vesicles and sent to Golgi apparatus. So that is correct. Now Clathrin coated vesicles are primarily involved in vesicle trafficking between the Golgi apparatus and endoplasmic reticulum. No. Between Golgi apparatus and endoplasmic reticulum, it is COP1, not Clathrin coated. Clathrin coated, I already told you, mediate transport originating in the Golgi apparatus to endosomes and then to plasma membrane. So E is not correct. So that means A, C, D are correct, and that makes option 1 as your answer. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Thank you.